Howdy folks, thanks for checking in to Mr. Ulrich's Landobiology.com. I am Mr. Ulrich. In this video, we're going to be looking at the nitty-gritty of molecular genetics. We're going to be looking at DNA, RNA, proteins, and the processes that go between these different compounds. In the beginning of a genetics unit, I will often ask my students, what is DNA? When we're studying cells, I'll often ask them what's the nucleus for, and most students will proudly say that the nucleus is the control center of the cell, and the DNA tells the cell what to do, or the DNA is what makes me who I am. And those are generally not incorrect. They do kind of gloss over some of the important parts of DNA and how it actually works to control the cell. And so we're going to dig a little deeper into this here. You may remember we talked about way back in the beginning of the school year when we were talking about proteins. I introduced this very important flow chart where we started out saying that the base sequence of DNA in a gene, and we're talking about the bases, we're talking about those A's, T's, G's, and C's. All right, the base sequence of the DNA in a gene, which of course lives in the nucleus, that is directly influencing the amino acid sequence of a particular protein. There are 26 different amino acids, and they have to be arranged in a particular order. And this arranging of the amino acids in an order to make a protein takes place at the ribosome. So the particular amino acid sequence that a protein has will determine the shape of that protein, and then the shape of that protein is going to determine the function of the protein. And I hope you remember when we were talking about proteins how many different functions different proteins have, from hormones and neurotransmitters to receptor molecules to enzymes. These guys are really, really important. Proteins are the workhorses of life. They really make life possible. And of course, it's DNA that gets all the credit. And hey, DNA, it's important stuff. It is the instructions for protein construction. But we got to give the proteins their due. So in this little video, what we're going to be looking at, we're going to be focusing on how we go from the base sequence of DNA in a gene to the amino acid sequence of a protein. And the processes that are involved are called transcription and translation. So it's not the DNA that's doing the direct controlling of the cell, it's the proteins that are actually doing the controlling of the cell. And this graphic, in fact, quite a bit of the graphics that I got, some of the text that I got from this whole PowerPoint here, I got from a teacher in New Jersey, Liz LaRosa. This pretty good graphic shows how the DNA codes for those proteins, going from DNA through transcription into RNA, and then from RNA through translation into proteins. Those structural proteins and enzymatic proteins and hormonal proteins are the things that are going to go out and cause the physical traits and influence the metabolic traits and control growth and development. So we got a basic problem. The information for making proteins is located in the nucleus, tied up in the DNA. The place where the proteins are actually going to be assembled, well, that's in the ribosome, not in the cytoplasm. So somehow we got to get the information, the instructions, from the nucleus to the ribosome. And that's going to be the job of ribonucleic acid, okay, better known as RNA. Messenger RNA is one type of RNA. We're going to meet a whole bunch of other types later on. But messenger RNA, its job is to bring the code of DNA from the nucleus to the ribosome. It's very similar to DNA. It uses a sequence of nitrogenous bases to encode information, and the nitrogenous bases are almost exactly the same. It's A, U, G, and C. That's adenine, uracil, guanine, and cytosine. So it's almost the same as DNA. The only difference base-wise is instead of having thymine, the T, it's got uracil, U. So the messenger RNA is kind of the PDF or portable document file of protein synthesis. It's what's going to carry the information from one place to another. Like I said, there are other types of RNA. We'll get to those as we get further on. The first step in going from the base sequence of DNA to the amino acid sequence of a protein is called transcription. Transcription is that process of copying the DNA's code into messenger RNA. And of course, in order for that to happen, the DNA first has to unzip, and then the messenger RNA is going to copy that DNA using the same base pairings. Now, like we said, RNA is a little bit different from DNA. It's single-stranded, not double-stranded. Instead of deoxyribose as a sugar like in DNA, it has ribose. And like we said before, instead of using thymine like DNA, it uses uracil instead. The job of messenger RNA is to carry that genetic code from the nucleus to the ribosome in the cytoplasm. So here's an artist's rendition of transcription, the first step of transcription anyway. Thank you, Mrs. Costello. Here you can see the DNA has unzipped the DNA template, and individual messenger RNA nucleotides are coming in and bonding, hydrogen bonding with the bases of DNA, using the same corresponding bases, keeping that same code, G's to C's, A's to U's in this case, or T's to A's. So we're converting that DNA base sequence into a base sequence of messenger RNA. It's still bases, so it's the same language, and that's why we call it transcription. Transcription literally means to just copy something over from one place to another, just like we would do in music. If you're changing keys or changing instruments, it's still notes when you transcribe from one key to another. They're the same intervals, it's just different note names, I guess. And this, of course, is occurring in the nucleus. So once that messenger RNA strand has formed, it starts to work its way through pores in the nuclear envelope into the cytoplasm, where it's going to bond with a ribosome. Now that the messenger RNA is at the ribosome, we can start going from the base language, the base sequence language of nucleic acids, to the amino acid sequence language of proteins. Now, just like our alphabet has 26 different letters that we can arrange in different ways to make all the different words that we know of, there are about 20 amino acids in the protein language, and we can arrange those amino acids in different ways to make different proteins, to make all the different proteins that we know. Now, the problem in converting from the A's, T's, G's, and C's to, or the A's, U's, G's, and C's, now that we're dealing with messenger RNA, to all of the different 20 amino acids is that there's just not enough. There's just not enough bases. So we can't have one base equals one particular amino acid. We can't even have a certain combination of two bases equals an amino acid, because there's not enough combinations of pairs of those four different bases. What we do have, though, are triplets. There are more than enough triplet combinations of those four bases to account for all the different amino acids that there can be. These three base sequences that correspond to particular or specific amino acids are called codons, and each codon codes for one of the 20 different amino acids. So the codons make up the template for protein synthesis, and that protein synthesis is going to take place at the ribosome in the cytoplasm. So what we're talking about here when we're going from one language to another is translation, and that's what we call going from the nucleic acid language of bases to the amino acid language of proteins. It's called translation. So in translation, the ribosomal RNA, which is RNA that makes up the ribosome, attaches to the messenger RNA, and there's enough space on that ribosome for two codons to fit. Now, transfer RNA, which is another type of RNA, it's attached. Each transfer RNA has an amino acid attached to it, and that particular transfer RNA will only fit with one type of amino acid. And on the other side of that transfer RNA is what we call the anticodon. It has the corresponding three bases that will fit with the codon that's on the messenger RNA. So as the transfer RNAs come to each codon, they add a amino acid until the chain of amino acids is complete and we have a functioning protein. So here's another artist's rendition of the process of translation. Thanks again to Mrs. Costello. It's a little easier to see here. The two kind of semicircles in the middle there, that's the two subunits of the ribosome. There are two subunits of the ribosome. One's bigger than the other, and that's why it looks kind of like a lopsided peach or something like that. Going through the middle there with the GGG, UAU, GGU, CCU, AUG. All right, that's the messenger RNA strand, and you can see where there are two codons in position, held in position in that ribosome. And then there are the two transfer RNAs with their anticodons joining up with the complementary codons that are in the ribosome. And those transfer RNAs are attached to their specific amino acids. That's what those shapes are. You can even see other transfer RNAs with their amino acids floating around in the cytoplasm. Once those two transfer RNAs are bonded temporarily to their codons, a peptide bond will form between the two amino acids. A little energy is utilized, and the whole thing shifts one codon. And the transfer RNA that was in the ribosome but is no longer breaks off and goes to complex with another amino acid, and that amino acid that's attached to the neighboring transfer RNA sticks in position. So the messenger RNA base sequence is converted into the amino acid sequence. We are dealing with a change in the language, just like we would in music, and this occurs at the ribosome. So the process is going to continue with the ribosome moving along the messenger RNA strand, one codon at a time, the transfer RNA bringing in the next appropriate codon and adding that to the growing chain of amino acids. We're going to call that chain of amino acids a polypeptide, poly for many. And for our purposes, we're going to equate polypeptides with proteins. There is difference. Once that polypeptide bends into a particular shape and interacts with other polypeptides is when we're going to really call it a protein. But for our purposes, polypeptide, protein, same thing. Well, let me tell you, if ribosomes, which are non-thinking blobs of RNA, are capable of recognizing individual triplets of bases, those codons, and converting those to individual and particular amino acids, well, you can too. It's easy once you know how to use the handy-dandy messenger RNA codon translator table. Let's say we have a triplet, UAU, and we want to figure out what amino acid that corresponds to. The first nucleotide column there tells us which row to look in, and since our first nucleotide is U, we're going to be in that first row. The second nucleotide tells us which box within that that row that we're going to be looking at. And since our second nucleotide is A, it's going to be in that particular box right there. Now we just got to figure out which line within that box we're looking at, and that's going to be the third nucleotide that's going to tell us that. Since it's a U, it's the top one, and we're looking at TYR, better known as tyrosine. So here's a couple other ones. Here, check this one out. If you want to hit pause, you can hit pause. All right, that's proline. Take a look at AGU. You can hit pause. That's serine. How about GCA? Once again, hit pause. All right, that's alanine. All right, you can see in this list there is some redundancy. Right? If you look at GUA, GUC, GUG, GUU, they're all the same. They're all val or valine. Right? And that's kind of handy. What that means is that if there's a mistake, if there's a mutation, and there's a change in that third nucleotide, it's still going to code for valine, and you're not going to have a change in the protein. So that's kind of that redundancy allows there to be some mistakes made and not have it be totally horrible. Well, here's a summary in picture form of the whole process. All right, so we got DNA up at the top. That unzips and is transcribed into a messenger RNA strand, which moves from the nucleus to the ribosome, where it is translated into a particular order of amino acids. And it's that order of amino acids